Hi, thank you for joining me today. My name is Nicole Gentili, and I'm a graduate student with the UC Davis Lawrence Livermore National Laboratory Point of Care Technology Center. Our center's website and email address are listed here for correspondence. Our focus is pathogen detection, which is directly related to today's topic of discussion titled Assessing Performance and Validating Point of Care Testing, Challenges with Nucleic Acid Recognition. My goal today is to provide you with an evidence-based overview of the concepts involved with assessing performance of nucleic acid recognition devices, the need for nucleic acid recognition at the point of care, comparisons and potential limitations of nucleic acid recognition, validation principles, as well as confounding factors and challenges associated with validation. Before I begin discussing the core content of the lecture, I would like to define some concepts you may hear throughout the talk. Evidence-based medicine is defined as the conscientious, explicit, judicious use of the current best evidence gained from the scientific method in making decisions about the care of individual patients. I would like to explain how to interpret test results in an evidence-based manner. This 2x2 contingency table defines true positive, true negative, false positive, and false negative results. A test result is considered a true positive result when an infection is reported and, in fact, is present in the individual, i.e., a positive test result in a patient who is disease positive, as represented by the TP in the table. A test result is considered a true negative when an infection is not reported and, in fact, is not present in the individual, i.e., a negative test result in a person who is disease negative, represented by the TN in the table. A false positive result occurs when an infection is reported but is absent in the individual, while a false negative result occurs when an infection is not reported but is actually present in the individual. A nucleic acid recognition test is designed to maximize the true test results while minimizing the false results. The next concept I want to discuss is the analytical performance of a nucleic acid recognition device, which is assessed via sensitivity and specificity. Analytical sensitivity is defined as the lowest concentration of target nucleic acid that can be reproducibly measured. Another name for analytical sensitivity, which you will hear very commonly, is limit of detection, or LOD. Analytical specificity is defined as the method's ability to obtain negative results in concordance with a negative reference result. In other words, it is the degree of which the assay detects the specific target and does not produce false positive results in the presence of non-specific targets. Analytical sensitivity is reported in units such as DNA copies per mil or colony-forming units per mil, while analytical specificity is reported as a percent. Clinical utility of a nucleic acid recognition device is assessed via sensitivity and specificity as well. Clinical sensitivity, or true positive fraction, is defined as the proportion of patients known to be positive for a disease whose values test positive at a particular decision threshold. It is the ability of the test to correctly identify a disease. Therefore, with a highly sensitive test, a negative result will allow you to rule out the disease. This can easily be remembered by the word SNOUT, the SN representing sensitivity and OUT for ruling out the disease. Clinical specificity, or true negative fraction, is defined as the proportion of healthy subjects whose test results are negative or within the defined decision limit. It is the ability of the test to correctly identify the absence of disease at a particular decision threshold. Therefore, with a highly specific test, a negative result will allow you to rule in the disease. This is remembered similarly by SPIN, where SP represents specificity and IN for ruling in the disease, SPIN and SNOUT. Both clinical sensitivity and specificity are reported as a percent. Post hoc analysis design and analysis of experiments refers to looking at the data after the experiment has concluded. The positive predictive value, also known as precision rate or post-test probability of disease, is the proportion of patients with positive test results who are correctly diagnosed. It is one of the most important measures of diagnostic utility as it reflects the probability that a positive test reflects the underlying condition being tested for. The negative predicted value is the proportion of patients with negative test results who are correctly diagnosed. Our first topic of discussion will be the need for nucleic acid recognition at the point of care. Point of care testing is defined as diagnostic testing at or near the site of patient care. Nucleic acid recognition assays represent molecular techniques designed for detecting pathogens. Pathogens include bacteria, such as gram-positives and negatives, viruses, fungi, and parasites. This slide illustrates some examples of nucleic acid recognition devices. The three categories represented are laboratory tests, shown in the left red portion, near-patient tests in the middle yellow portion, and bedside tests in the right green portion. Near-patient and bedside nucleic acid recognition tests represent point-of-care tests. I want you to focus on the right green portion. There are currently no nucleic acid recognition devices for hospital use at the patient's bedside. The only approved handheld device for pathogen detection is a BioSeq device used for detection of biothreats. It is not used for patient samples and hence is not shown here. Developing bedside nucleic acid recognition tests is a current focus in the point-of-care testing field, which will result in emerging technologies for the years to come. Nucleic acid recognition at the point of care is important. Recent studies have demonstrated that point-of-care nucleic acid recognition tests, particularly PCR-based assays, have the potential to enhance detection of pathogens. This rapid diagnosis of pathogens could lead to early evidence-based treatment decisions. Additionally, novel point-of-care nucleic acid recognition devices may facilitate diagnosis and treatment in critical, emergency, and disaster care settings. I am now going to provide you with an example to demonstrate the value of a highly sensitive point-of-care nucleic acid recognition test. Susan Fiskus and colleagues published results in 2006 from a prospective study designed to evaluate HIV detection assays in a high-prevalence population in Africa. They calculated that 70 to 95% of acute HIV infection were missed when solely based on rapid antibody test. The rapid antibody test happens to be the initial testing method recommended by the Center for Disease Control and the World Health Organization. But this 70 to 95% is huge, especially in a high prevalence population. The research team claimed that the sensitivity of the initial rapid antibody method would be substantially increased if a nucleic acid recognition device were used in parallel. Pilcher and colleagues successfully demonstrated this technique in the United States by combining a pool of antibody-negative samples with a nucleic acid recognition method to aid in the diagnosis of acute HIV infection. This figure illustrates the current guideline for HIV testing from the Center of Disease Control and Prevention. The initial method for testing for HIV is a rapid antibody method, as mentioned previously. An example of this form of testing is listed on the right, which is the OraQuick Advanced Rapid HIV 1 and 2 antibody test with a documented sensitivity and specificity of 99.3% and 99.8% respectively. If this initial test result shows a positive result following the arrow pointing down to the left with the green plus sign, a second test is required to confirm diagnosis. This second HIV test can either be another rapid antibody test, like the initial test, or a nucleic acid recognition test. If this second test result is positive as well, the patient is considered to be HIV positive. Final diagnosis. If the second test result is negative, however, a third test is still needed for confirmation. This third test can also be a rapid antibody test or a nucleic acid recognition test. The third test result will then be the deciding factor of whether the patient is HIV positive or negative. The question I pose to you now is, will this patient who had 3 HIV tests performed going to be strongly convinced by the final diagnosis? Now, let's consider another completely unrelated example of a patient being tested for HIV. Returning to the top of the figure and following the arrow pointing down to the right this time with the negative sign. This is a patient whose initial rapid antibody result is negative. According to the CDC, this negative result is indicative of an HIV negative diagnosis. There is a potential problem here, however. Although a sensitivity of 99.3% for the initial rapid antibody test sounds sufficient and wonderful, unfortunately, there will be a large amount of false negative test results produced in large populations with a high prevalence of HIV. This was particularly shown by the prospective experiment performed by Susan Fiskus and colleagues. Being that this initial negative test result is supposedly indicative of a negative diagnosis, this means that many truly positive HIV patients will be improperly diagnosed as negative. I created an example to demonstrate the severity and consequences of this issue, highlighting the value of highly sensitive tests. Okay, so sub-Saharan Africa had a rather large population of 800 million in 2007. Of that 800 million, approximately 3%, or 22 million people, were living with HIV or AIDS. Let's just say that every person in this particular population of 800 million was tested for HIV. Using an initial rapid antibody test with a sensitivity of 99.3%, as mentioned earlier, will lead to 0.7% of the population to be falsely diagnosed as HIV negative. Therefore, according to the calculation, 800 million times 0.007 equals 5,600,000 false negatives. That is a lot of people who will not receive proper counseling and treatment for this severe disease in the beginning. In conclusion, I propose that using a more highly sensitive, rapid, and inexpensive nucleic acid recognition device as the initial HIV test will decrease the incidence of false negative results in high prevalence populations such as sub-Saharan Africa. Unfortunately, though, there are sensitivity and specificity trade-offs, meaning that the two really are inversely proportional. Increasing the sensitivity of a test will generally decrease the specificity. According to the equations shown, sensitivity and specificity are associated with false negative and false positive results, respectively. Therefore, increasing the sensitivity will lead to a decrease in false negative results. Unfortunately, this decrease in false negatives will likely lead to an increase in false positive results. Conversely, increasing the specificity will lead to a decrease in false positive results with an associated increase in false negatives. I will provide an example to visually demonstrate this in the following slide. Okay, this slide may appear complicated at first sight, so let me break it down for you before explaining the key concept. The left panel represents detection of disease by a highly sensitive and less specific test, while the right panel illustrates detection with the less sensitive, highly specific test. The X and Y axes on each graph represent increasing analyte concentrations and seroprevalence of herpes simplex virus. The green and red curves illustrate HSV negative and positive populations. Almost always there is some degree of overlap between results from normal and diseased subjects. For example, here low levels of blood markers of HSV may be detected in healthy subjects. These may overlap the lower range of values found in patients with HSV. When there is such an overlap, some cutoff point must be established to distinguish a positive from a negative result. This is where desired sensitivity and specificity come into play. The colored blue and orange areas under the curves represent false negative and false positive results given the sensitivity and specificity cutoffs, represented by the vertical black lines on the plots. The yellow arrows represent a shift of the black vertical lines from the highly sensitive test to a highly specific test, i.e. the trade-off. So here we see that shifting the black line from the 99% sensitivity, 95% specificity cutoff in the upper left graph to the 95% sensitivity and 99% specificity cutoff in the right upper graph produces a larger blue area under the curve. This represents an increase in false negative results. Clinically speaking, this could mean that HSV positive patients with analyte concentrations in the lower levels are actually misdiagnosed as HSV negative more often. We also see that shifting the black line from the 99% sensitivity, 95% specificity cutoff in the lower left graph to the 95% sensitivity, 99% specificity cutoff in the lower right graph produces a smaller orange area under the curve. This represents a decrease in false positive results. Clinically speaking, this could mean that HSV negative patients with analyte concentrations in the upper levels of the negative spectrum will be diagnosed as positive less often and properly diagnosed as HSV negative more often with a highly specific test. I hope this graphical illustration is a helpful tool for explaining the sensitivity and specificity trade-off in regards to false positive and false negative results. Our next topic of discussion will be limitations and comparisons of nucleic acid recognition tests. Besides the sensitivity and specificity trade-off phenomenon, nucleic acid recognition unfortunately exhibits some potential limitations, some of which are listed here. For example, it is possible that cross-reactions can occur with multiplex detection of pathogens. More reactions means more primers and more interference. It is possible that primers can interact with each other and interfere with pathogen detection. Also, nucleic acid recognition methods requiring extraction steps demonstrate potential for higher false positive rates due to low-level contamination. False positive results may also occur if the number of target sequences in a sample is small and degradation of the DNA or RNA takes place. In samples containing large amounts of human DNA, non-specific amplification can occur when the unrecognized DNA sequences are similar to the target sequence of the organism in question. Additionally, erroneous results may occur for many reasons, such as non-specific primer binding and amplification of other sequences that are misidentified as the target sequence, amplification of similar target sequences found in other organisms, and low levels of contamination. The latter may be caused by residual products from the previous reaction causing what's called amplification carryover. Many of these issues can be overcome with proper primer design and decreasing the complexity of the nucleic acid recognition instrument. For example, a less complex point-of-care nucleic acid recognition system with limited operator handling will likely have less contamination issues than a complex laboratory-based nucleic acid recognition system requiring extensive operator handling and processing steps. This slide compares laboratory-based nucleic acid recognition assays with point-of-care assays. One major issue with current laboratory pathogen detection methods involves pre-analytical processing or sample preparation steps such as cell lysis and extraction. The tedious hands-on techniques required during pre-analytical processing stages, such as required with PCR, may encompass approximately 40 to 50% of the total processing time. That is a lot. Total processing time is defined as the total time required for sample preparation, reaction, and analysis. In addition, DNA is effectively on every surface and any solution in our environment. This may pose as a source of contamination for nucleic acid recognition devices during the tedious hands-on steps previously mentioned. I also mentioned earlier that some multiplex detection capabilities may be hampered by DNA contamination. To assist in solving this problem with PCR, for example, Roche developed molecular-grade or M-grade DNA-free products where the DNA fragments are so small so as not to cause background noise and threshold detection by the device. A better solution, however, would be to eliminate the pre-analytical processing steps altogether by developing a point-of-care nucleic acid recognition device that performs automated pre-analytical processing steps inside the system. This effectively will decrease the chances of ubiquitous DNA contaminating the reaction mixture and will in turn decrease the false positive results. This slide is designed to further illustrate the comparison of total processing time between laboratory-based nucleic acid recognition testing, represented in the top red box, and point-of-care-based nucleic acid recognition tests, represented in the bottom green box. Again, total processing time is defined as total time required for sample preparation, reaction, and analysis, or the total time required to complete pre-analytical, analytical, and post-analytical processing steps. The total processing timeline is indicated by the horizontal white lines with tick marks. Each tick mark indicates 1 hour. Pre-analytical processing time is represented as the pink bar on the timeline, analytical processing time in the blue bar, and post-analytical processing time in the yellow bar. Notice that the pre-analytical processing stage of this particular laboratory-based PCR test encompasses approximately half of the total processing time. This is due to the complex and tedious hands-on techniques. The actual analytical reaction time also encompasses a large portion of total processing time because it utilizes a thermocycling method. Total processing time of this particular laboratory test is between 5 and 6 hours. Not very ideal for disaster situations or emergency and critical care for that matter. Total processing time for a point-of-care nucleic acid recognition test ideally should be less than 1 hour to be useful for critical emergency and disaster care settings, where rapid diagnosis has great potential for improving patient care. The bottom green box represents a design of a point-of-care nucleic acid recognition device called loop-mediated isothermal amplification, or LAMP. This particular device is currently being developed by our center. Our goal is to make it less complex when compared to a laboratory-based method. For example, the sample will be collected and directly placed into a device where pre-analytical processing steps are minimal or absent and require minimal operator handling. The analytical and post-analytical processing steps will be decreased as well. The analytical processing time will be decreased in this particular method by utilizing an isothermal amplification approach, where no temperature cycling is required, like PCR. The post-analytical processing time is minimized by the quick and easy-to-read results screen. In this particular example, the LAMP is positive for the SPA gene and negative for the MEK-A gene, indicating a positive Staphylococcus aureus result, but negative for MRSA. I will explain this in more detail in a few slides. Point-of-care testing must meet the minimum standards specified in Clinical Laboratories Improvement Amendments, also known as CLIA, of 1998 regulations. The CLIA categories include wave testing, moderate complexity, and high complexity. CLIA wave tests employ methodologies that are so simple and accurate that it would render the likelihood of discrepant results negligible. Also, if for some reason the tests were performed improperly, no harm would occur to the patient. Moderately complex and highly complex tests are scored based on a variety of criteria. These scores depend on the level of knowledge of the operator to perform the test, training and experience, reagents and material preparation, characteristics of operation steps, calibration, quality control, and proficiency testing materials, as well as test troubleshooting, maintenance, interpretation, and judgment. This slide compares pathogen detection methods relative to their level of complexity based on CLIA definitions. From left to right, the complexity decreases. Panel A shows a highly complex test requiring molecular diagnostic lab. Panel B shows a cartridge-based test with moderate complexity, and Panel C shows a CLIA-waived whole blood non-nucleic acid recognition test, which can be used by most minimally trained users. There are currently no CLIA-waived approved nucleic acid recognition tests available, unfortunately. Many nucleic acid recognition systems are classified as open systems. Utilizing an open system means that rather than immediately placing the sample into a self-contained unit for processing, the sample is processed outside of the testing system first. The increased environmental exposure may influence the end result. In addition to the biosafety hood required for sample preparation, an open system requires an additional separate room for the actual reaction, whereas semi-closed systems do not. The tedious sample preparation techniques for an open system reaction require extensive operator handling, which increases the level of complexity and time to result when compared to the closed system. The decreased operator handling and complexity of semi-closed systems are characteristics which allow them to be classified as moderately complex or CLIA-waived, rendering them candidates for point-of-care use. This slide illustrates an open system on the left. Here, letter A is the Roche Septifast Light Cycler PCR, versus a closed system on the right. Here, letter B is the Cepheid Gene Expert, with their corresponding test clusters shown in white. Again, an open system requires a biosafety hood for performance of pre-analytical processing steps. Once the extraction and lysis steps are performed, reagents along with extracts and master mix are loaded into a carousel. The reaction is performed in a separate room. These manual methods may expose samples to contamination from ubiquitous organisms in the environment. Manual processing may also be vulnerable to user error. More recent tests, such as the Cepheid Gene Expert, utilize self-contained reagent and reaction packages which reduce the potential for contamination. The Gene Expert system is currently the only near-patient PCR-based pathogen detection system with a semi-automated cartridge-based design. The sample is collected and then placed into a cartridge. Reagents are added and the system's automated microfluidics perform the pre-analytical processing steps internally. This nearly eliminates the potential for contamination and in turn will decrease the incidence of false positive results. Despite whether the reaction is considered an open or closed system, CLIA-waived or complex, nucleic acid recognition devices, like all other molecular diagnostic tests, can fail if not developed or maintained according to the proper standards. Failure of a molecular diagnostic device may lead to misdiagnosis or inappropriate and/or inadequate treatment decisions. I'm now going to discuss with you the principles of validation, which are designed to ensure the device complies to proper standards. Validation is a process to confirm that a nucleic acid recognition test is performing properly, complies with design standards, and demonstrates reproducible results. The validation process encompasses both analytical and clinical performance experiments. These experiments are designed to confirm that the assay is specific enough to detect the claimed target only, for example, all Staphylococcus aureus strains, and will not detect related species, such as Staph epidermidis. Therefore, a highly specific nucleic acid recognition assay will ensure that there are minimal false positive results produced. Optimal primers must be designed so as to pinpoint detection of the correct pathogen genes in question. Validation experiments ensure that this occurs. This complex Venn diagram I'm going to try to break down for you is designed to explain detection using Staph aureus as an example. The large left SPA positive circle illustrates all staph strains carrying the SPA gene, which is a protein A encoding gene specific for Staphylococcus aureus. The large right MEC-A positive circle illustrates staph strains carrying the gene for methicillin resistance. The Van-A positive circle represents staph strains carrying the gene for vancomycin resistance. A nucleic acid recognition device should be able to detect all strains of Staph aureus, including resistant strains such as vancomycin-resistant Staph aureus, also known as VRSA, and methicillin-resistant Staph aureus, or MRSA. This means that the assay should detect all species represented in the entire left circle. This encompasses the green, yellow, and pink portions. The assay should not detect any related neighboring species, such as those listed in the right orange portion. Less is known about vancomycin resistance than methicillin resistance, so current nucleic acid recognition assays detecting the Van-A gene are non-existent. So let's take, for example, an assay designed to detect the SPA and MEC-A genes. This nucleic acid recognition test could report an SPA positive MEC-A negative result, which corresponds to methicillin sensitive Staph aureus, shown in the green. Or the assay could report an SPA positive, MEK-A positive result, which would correspond to MRSA, shown in the yellow. However, since this particular assay example is not designed to detect the VAN-A gene, you would not be able to decipher whether this SPA positive, MEK-A positive result represented MRSA or if it truly represented a VRSA strain, shown in the pink. Ideally, in the future, the assay should be able to differentiate between all non-resistant strains and all resistant strains of Staph aureus. This discrimination is clinically important because it will ultimately impact treatment decisions. Nucleic acid recognition devices are classified as in vitro diagnostic medical devices because they are used to test patient samples for diagnosis of pathological, physiological, or therapeutic states. All IVDs are regulated by the IVD Directive 98-79-EC that entails a validation requirement for all IVDs. It also states that the purpose of validation is to prove diagnostic capability of the device and to ensure that the IVD does not compromise the care of the user. The Clinical Laboratory Improvement Amendment, or CLIA 88, mentioned briefly earlier, is required for verification and validation. The amendment was created to establish quality standards for all laboratory testing to ensure accuracy, reliability, and timeliness of test results. Tests can be waived from regulatory oversight if they meet certain requirements established by the CLIA 88 law. Currently, there are more than 100 CLIA waived tests available, not nucleic acid recognition tests, though. Waived tests are important because they are convenient, efficient, and rapid, 3 factors that improve patient care. CLIA regulations must be satisfied before the test result can be used to influence patient care. The companion to the IVD Directive 9879-EC is the International Organization for Standardization, or ISO 15189. This standard is the successor of ISO-IEC 17025 of 1999, which originally stated that the general requirements for the competence of testing and calibration laboratories. ISO 15189 is the main and current standard for quality and competence in medical laboratories. It states that analytical performance of clinical laboratory testing should be evaluated against objective quality specifications. For example, external quality assessment determines how a particular IVD is performing in comparison to similar IVDs, which is key, or other laboratory tests such as gold standards. The ISO 22870 specifies requirements applicable to point-of-care testing and is used in conjunction with ISO 15189. Analytical performance assessment should include bench experiments which validate the nucleic acid recognition device against known inhibitors and potential confounding factors. We define confounding factors as compounds which impair an assay to the point of causing discrepant and/or erroneous results. Confounding factors may be endogenous or exogenous in nature. Many of these confounding factors are overly expressed in critically ill patients due to medical and/or surgical interventions in various disease states such as sepsis. All known and newly discovered confounding factors and inhibitors need to be documented before the device can be released out into the field. These next 2 slides illustrate examples of potential confounding factors of nucleic acid recognition. The confounding factors are represented with pictures, and the corresponding information is represented in the text. On this slide, I want to focus on the blue text representing 2 well-known endogenous confounding factors of nucleic acid recognition. White blood cell genomic DNA is a well-known inhibitor of nucleic acid recognition. A white blood cell count of 300 to 30,000 cells per microliter serves as the operational range of many nucleic acid recognition tests, such as Septifast by Roche. Exceeding these limits may lead to false negative results due to dilution of the target DNA or sequestration of target DNA, respectively. The encountered white blood cell count in critically ill patients varies based on the etiology and pathophysiology of the disease state. AIDS patients, for example, may exhibit leukopenia where white blood cell counts are less than 400 cells per microliter, and septic patients may experience leukopenia and/or leukocytosis being greater than 12,000 cells per microliter. The white blood cell count may also vary depending on when one collects the blood sample and if surgical procedures have been performed. Blood urea nitrogen, or BUN, is also a known inhibitor of PCR. Severely septic patients or patients experiencing septic shock experience organ dysfunction, for example, kidney failure. Patients with acute renal failure will present with an elevated BUN, which may inhibit pathogen detection. I briefly just want to mention two other endogenous substances of which may pose as confounding factors. DNA-degrading enzymes like DNase and other nucleases present in serum samples may influence the half-life of circulating DNA. Also, patients exhibiting acidosis or alkalosis may expose DNA polymerases to denaturing pH levels. Taq polymerase operates optimally at pH of 8.3, but patients who have respiratory or metabolic acidosis or alkalosis may have blood pHs exceeding the operational range of PCR polymerase. Although these two potential confounding factors in the black text can potentially pose as threats to pathogen detection, less is known about the extent of interference or inhibition on nucleic acid recognition. Now I want to focus on two well-known exogenous confounding factors, again represented in the blue text. The first is ethylene diamine tetraacetic acid, or EDTA, which is a divalent cation chelator. Therefore, magnesium may be removed from solution and inhibit DNA polymerase, which depends on magnesium. Inhibition occurs at concentrations greater than or equal to 0.5 millimolar. Specifically, there has been observed inhibition of Streptococcus pneumoniae, PCR, from whole blood with EDTA. The clinical significance of this inhibition is that EDTA is present in some vacutainers that are used for blood sample collection. The blood will be mixed with the EDTA in the vacutainer and will then be subject to testing by the nucleic acid recognition instrument. Intriguingly, though, it is important to note that although EDTA is a known inhibitor of PCR, it also abolishes DNase activity, thus enabling successful amplification of microbial DNA after storage of EDTA anticoagulated blood samples. Just an interesting note. The next exogenous confounding factor to focus on is user error. Improper dosing and improper sample collection by the user may cause contamination and predispose the assay to erroneous results. Poor cleaning of the sample collection site or sample collection container could lead to contamination. Contamination may also come from poor sample collection techniques or improper selection of the sampling site. Now I briefly want to mention two other exogenous confounding factors which may pose as a threat to nucleic acid recognition, shown here in the black text. Transfusions using packed red blood cells, fresh frozen plasma, and platelets are often used in surgical patients to maintain blood pressure as a result of sepsis or blood loss. Fresh frozen plasma and platelets are also used to treat some coagulopathic states. Also, patients experiencing severe sepsis may require fluid resuscitation to maintain their blood pressure. Therefore, the addition of other fluids, such as saline or albumin, may dilute pathogen levels found in the blood. This table lists some known inhibitors of PCR in particular, besides the common inhibitors EDTA and white blood cells. The column on the far left lists the source of inhibitor. The middle column lists the inhibitor name, and the far right column describes the clinical relevance of the inhibitor. The common anticoagulant heparin is a known inhibitor of PCR. The exact mechanism is not known, however. There is possible interaction between PCR master mix and the highly anionic sulfated side chain of heparin. Critically ill patients may be on high doses of heparin. Therefore, this may inhibit PCR or other nucleic acid recognition techniques. Some other PCR inhibitors may be found in critically ill patients' blood at abnormal levels. These inhibitors include immunoglobulin G, or IgG, which inhibits the DNA polymerase required to amplify the pathogen DNA for detection. Hemoglobin found in red blood cells may release iron when lysed. Iron also inhibits the DNA polymerase. And heme found in hemoglobin, which inhibits amplification by altering diffusion rates of macromolecules in the reaction mixture. The last inhibitor I want to mention is the muscle tissue breakdown product called myoglobin. Myoglobin is a potent inhibitor of DNA polymerase, specifically Taq polymerase. Patients with compartment syndrome may have elevated myoglobin in their blood. Additionally, myoglobin is harmful to the kidneys, leading to elevated BUN levels, secondary to acute renal failure. You may recognize this from the previous slides. Inhibitors can have differential effects that are concentration dependent. The concentration effect is particularly relevant when the actual inhibitor concentration is usually unknown. Inhibitors may have unequal effects on different PCR protocols. For example, one particular PCR reaction may be unaffected by a potential inhibitor, whereas another may be completely suppressed. Given this notion, though, validating a new nucleic acid recognition device, considerations must be taken to understand the similarities of the intended reference method with the experimental method. This leads us into some potential challenges that will be encountered with both analytical and clinical validation of a novel nucleic acid recognition device. The next two slides will focus on challenges associated with analytical validation. It is well known that standards and controls are a routine part of laboratory practice. However, problems arise when values are not reported in standard units and when standards and controls are not readily available, which unfortunately is the case with most nucleic acid recognition assays. For example, results can be reported in DNA copies per mil, colony forming units per mil, plaque forming units per mil, DNA copies per reaction, particles per mil, the list goes on really, which leads to difficult interpretation of results. There is no easy conversion from DNA copies per reaction to CFUs per mil, which CFUs per mil is really common. The conversion needs to account for how many copies of repeated targets are found in the genome of the organism. The lack of standard unit reporting can pose a problem during point-of-care connectivity and transfer of results from one physician or hospital to another if each physician does not utilize the same reporting units. Ultimately, this may affect patient care and management. A second challenge is that standards and controls are not readily available for nucleic acid recognition of many organisms, especially uncommon pathogens. The only existing definitive standards for nucleic acid recognition are for viral detection, which include hepatitis C virus, hepatitis B virus, and HIV. These standards happen to be reported in RNA copies per mil. To summarize this slide, nucleic acid recognition experiences a lack of standardization. In addition, results of routine measurements using point-of-care devices are usually standardized through calibration to a reference method. However, there is no universal harmonized reference method comparable to nucleic acid recognition tests. Therefore, when a reference system is lacking, results are only referred to a manufacturer's selected procedure and corresponding calibrator. This is a problem because calibration techniques may vary among manufacturers. The lack of a universal reference method impacts validation data analysis because different statistical methods are used to assess performance of the device. In summary, nucleic acid recognition lacks harmonization in addition to standardization. Challenges also exist that impact clinical validation procedures. The first challenge I want to discuss involves the lack of a comparable reference method, as mentioned earlier. Blood culture represents the current comparison method for identification of bloodstream infections with nucleic acid recognition. This poses a problem, though, because ideally molecular devices should be compared with other molecular devices, not a culture method, to obtain accuracy in results. This was stated by the ISO 15189 standard mentioned earlier. The molecular comparison method could be a method already in use at the respective hospital. However, again, there will be variations between devices and manufacturer calibration methods. Most hospitals use the blood culture method, but there are problems associated with blood culture as well. For example, blood culture is limited by existing antimicrobials, inoculum level, and pathogen growth characteristics. It also demonstrates a slow turnaround time, and although rigorous cleaning protocols have been established, contamination still occurs. However, blood culture does possess some clinical utility characteristics that nucleic acid recognition does not. For example, a broad, nearly comprehensive test panel. This leads us to the second challenge existing with validation of nucleic acid recognition, which is that a broad test panel is necessary to calculate predictive values of an assay. Predictive values are difficult to accurately report when comparing discrete versus non-discrete tests due to the lack of an all-encompassing test cluster. An example of a test with broad test cluster would be blood culture, and a discrete test would be PCR. The positive and negative predictive values can be calculated for each individual pathogen detected by a discrete panel, but not for the assay as a whole due to the limited test cluster. The third challenge deals with the analysis of validation data. It is critical that a sufficient number of clinical samples be tested to achieve statistical power when calculating sensitivity, specificity, and predictive values. Achieving statistical power is necessary to give credibility to the assay and to show reproducibility. It is important to encompass a broad patient population, for example, intensive care units, emergency room, general medicine wards. This is because the experimental patient population must be representative of the larger target population for the assay to be clinically useful. On the left of this slide is a picture of blood culture, which is a broad, nearly comprehensive test, while on the right is a picture of the septid-fast light cycler PCR made by Roche Diagnostics, which is a discrete test. The 25 pathogens detected by the PCR method are listed in the table. There will be 25 individual positive predictive values and 25 individual negative predictive values representative of the 25 pathogens detected in the discrete test panel of the septid-fast light cycler. A single positive predictive value and negative predictive value cannot be calculated for the device to represent bacteremia or fungemia, since other organisms may be present in the blood that are not present in the test cluster. Blood culture, though, is able to detect a broad scope of culturable bacteria. Therefore, a positive and negative predictive value for the blood culture test can be calculated for true bacteremia. The definitive predictive values are not exactly 100% accurate, though, because blood culture has difficulty growing fastidious organisms and some intracellular pathogens, as mentioned earlier. Clinical validation must encompass evidence-based methods of evaluation. As mentioned 2 slides ago, a broad patient population representative of the larger target population is needed to successfully demonstrate the true clinical utility of the assay. More often than not, this will require a multi-center trial. A multi-center study will not only provide valuable clinical data from a widespread geographical distribution, but will also demonstrate assay reproducibility. Now, validating assay results requires even further analysis and verification via arbitrated case review. This is a state-of-the-art approach where 3 or more arbitrators independently analyze all available evidence, including all test results, to determine the accuracy of the nucleic acid recognition result. Case review arbitration is a necessary benchmark activity which uses evidence to determine whether the nucleic acid recognition test result is accurate. In conclusion, challenges are associated with validation procedures. The analytical challenges include lack of standardization and harmonization for comparing and reporting results, as well as calibration procedures. Some clinical challenges involve the lack of a suitable universal comparison method, as well as the difficulty with calculating predictive values when limited to a discrete test panel. Validation of a novel nucleic acid recognition device is important to assess the analytical performance and diagnostic utility before releasing the instrument out in the field. Validation experiments are necessary to achieve the desired specifications of the assay and to identify potential confounding factors which may inhibit or interfere with pathogen detection. In particular, arbitrated case review is an essential part of assessing clinical utility. Additionally, potential limitations include possibility of false positive results due to contamination issues with highly complex assays. The increase in false positives will decrease the specificity of the device. This issue can be remedied through proper pre-analytical design. Therefore, moderately complex or CLIA-waived point-of-care nucleic acid recognition tests are needed. Validation must utilize an evidence-based approach, such as multi-center trials which will ultimately confirm the true value of pathogen detection at the point of care. Many of the challenges presented in the lecture can be overcome in the future. For example, I propose that instrument performance be validated against all potential confounding factors and that newly discovered confounding factors be properly identified before device release. Secondly, algorithms should be designed for detecting cross-reactions that may occur with multiple primers in multiple assays. Remember, multiplexed assays mean more primers and more inhibition potentially. Thirdly, a nucleic acid recognition universal comparison method should be developed in order to promote standardization and harmonization in the evolving field of molecular diagnostics. And lastly, complexity of nucleic acid recognition devices should be decreased towards satisfaction of CLIA-waived criteria. For example, pre-analytical processing steps should be less time-consuming and should require less tedious hands-on processes. The decreased operator handling will decrease the potential for contamination and thus decrease the incidence of false positive results, leading to more specific assays that can be utilized at the point of care. Thank you very much for joining me today. This lecture was supported by the National Institute of Biomedical Imaging and Bioengineering. The following reference lists will provide you with supplemental reading material.